basically, there's this inversion layer in the winter, and so it's just continuously socked in with clouds. And the moment you leave that valley, you get some sun, right? And so I'm told by people here that in non-corona times, you would just hop in your car, hop in your friend's car, and head outside of Ljubljana for a couple of hours just to see the sun, because the sun is quite a rarity here right now. But because the COVID restrictions are limited to our own municipalities, and so we're not allowed to travel just to go see things, see the sun. So Corona has stolen our sun as well. Anyway, it's yeah, it is what it is. So today I'm going to be again sort of thinking through some of these ways of representing the variants, the genetic variants, transgender experiences, and so forth in light of the conversations I had yesterday with Dr. Wong, and thinking about also within the broader theme of Lab and the part that I'm in of Forking Piragene, of forking some of his ideas into something else, and something that was very much unexpected potential new direction for this. I also wanted to say that later today at 3 p.m., 1500 hours, European time, Central European time, and 10 p.m. Taiwan time, there will be a special event called Body as Trans Data. And so this is an event with myself, a transgender woman programmer, free software activist, and an LGBTQ activist, Imacat from Taiwan. And from here in Ljubljana, there will be Leah Amard, who works for Transaxia, which is a local transgender group that does all sorts of advocacy and support. She's also a filmmaker and camera operator. And I've worked with a little bit with Leah during my time here in Ljubljana, having some discussions about hormones and transgender experiences and so forth. And so this will be a hour and a half long discussion and conversation between the three of us that will be streamed also on this channel. And people are welcome to join in the conversation and ask us questions and so forth, especially if you're transgender or non-binary. I also wanted to remind you all, if you're interested, of the Telegram group, Exomeopragmissions. If you're interested, come on, take a look. Yeah, so I think that's all for the announcements, I guess, of today. Let me just mute my mic while I take a sip of coffee. Okay, so let's think about what we're going to do today. I feel like to a certain extent, it looks like there hasn't been a lot of movement on the project, but I think conceptually there has been quite a lot. And so I think it behooves us to consider here a bit what types of representations are we thinking about with these genetic variants, and to sort of recap the sort of, to me, really sort of thought-provoking thing from Dr. Wong's conversation that I had with him yesterday, this idea that perhaps the genetic code and the variants that we find in it that are associated with transgender identity, such as in that study that I've been talking about a little bit that I was reading and was really fascinated by. So those variants perhaps don't have any kind of impact on sort of the phenotype of an individual, right? Perhaps not. We don't know. That's the problem. Not necessarily the problem, but the limitations of these studies, right? They're association studies. Um, they don't say anything about causation. They don't do anything about sort of like, you know, do these changes necessarily have a physical, biochemical, molecular effects on the body, at least in the types of studies like this one. Sure, if you're looking at genetic variants with respect to disease, then maybe you can have a better understanding that perhaps these variants are contributing to the disease. But even then, you don't necessarily know that. You have to do further studies to understand. So the study finds that there are these variants. So what's their purpose? What's their meaning? And for me, the really thought-provoking thing from the conversation was that Dr. Wong had suggested that maybe these variants are traces, deep historical traces, of a particular moments in time where certain populations had an ability to experience sexual identities, gender identity, sexual orientation in a much more expansive way. Maybe these societies were much more equal in this fashion. And thus, perhaps there was a trace that was left on the genetic code, in a sense. Perhaps sort of almost epigenetic trace. Um, so that might be an interesting thing to explore a little bit today, just a little bit on the research between, say, epigenetics and variants to see if there is anything. Um, and then, really thinking about, okay, we've been spending a lot of time looking at this VCF format and trying to fit things into that as a potential form of code poetry that can then be encoded in something to be transmitted. And perhaps that's a little bit convoluted of a way to think about this. So we'll think about that and see if there's maybe a different way to go about things. Um, and maybe also we can begin to upload some of this stuff to HackMD, which I haven't been doing, but I should. So maybe we can do some of that during the session today. And then also maybe begin to edit um, some of the website that runs the radio part of things in order to fork that and fragment it as well. Because I actually have forked the software on GitHub. And so, yeah, we can begin to think about that. And so, forking Piragene, we can think about this idea of fork as something that is, yes, drawn from computer technology, programming, and so forth, which suggests this idea of taking an existing code base within the context of programming and then deciding to take that code base into a different direction. So it's a fork. So the original code base is here, and then it forks off into another direction. Obviously, also the reference of the fork in the road. So the idea of taking something existing and breaking off from it and going off in another direction. And so I've forked this code on GitHub, and so we can also begin to edit some of this stuff and see where we can go with it. Originally, I had planned on starting to do some of the radio transmissions tomorrow. I think it might be best to continue this work session tomorrow, actually, and then try and start to do some of the radio transmissions on Friday. So let's maybe start doing some stuff with HackMD. And see about creating some pages on here where I can just sort of copy some of the things that I've been doing. Okay, cool. So let me just switch to my desktop work. Okay, cool. one more thing that I want to do is within the ticker, I just want to make a note about the body as trans data conversation. Body as trans data conversation today, 100 hours CET and 2200 hours CST. Okay. So let's do this. Get rid of that. Yep. And then let me just write something considering deep history as encoded in genetic variants. Cool. And then I just want to do something over here where I'm going to just quickly search on Google Scholar briefly so we can come back to this later. Genetic variants epigenetics. And I'll see if there's any sort of potentially research about this. Okay, yeah, cool. So, but for right now, let's go to the HackMD and start creating a new team note here. And what I'm going to do, excuse me, I'm going to copy all of this info that I've been working on. Let's see, I guess I'm going to have to maybe change some things around a bit. Let's see here. Yeah. Oh, yeah, sorry. I'm in a different sort of keyboard, so sometimes I forget how I'm supposed to do this in Vim. Ah. Okay, perfect. So great. Great. So this has everything. Cool. Let me just create another pound here and just say these are the Xomio missions daily notes. Cool. And then I can just copy and paste things. The links all seem to be working. Great, because it's Markdown. Wonderful. Okay, so does this just save? I sometimes forget. Just take a look at the help here. Don't necessarily want to lose all this, so let me copy this. And cool, okay. Let me check here. Yeah, so I just need to figure out how to tag it. How do I tag it? How do I tag it? HackMD tag entry. No, that's HackMud. Okay, you know what? We'll just leave it like this later. And we will just, yeah, come back to this later because we've got the daily notes. And yeah. Settings, no. Save your notes. There's probably some thing here to tag it. Anyway, we will yep, just let me take a look at something here. Right. Tags. Oh, how do we do this? Oh, there we go. That's how we do tags. Okay. So let me go back to my thing and tag it under the title. Cool. And then go back. Cool. And then let me put this under here and say, right, I need to get the link to this. And then change this. Link to it. Research. And then this. Daily notes. And then we do this. And does that work? Yeah, except for it goes into edit mode. So, sorry that I'm taking so much time on this, but yeah, I just want to, maybe I just need to go to the View tab. Okay, cool. There we go. That's good. That's good. Okay, so I think we're good there. We've uploaded some of that stuff, and so I think we're okay now. Close that tab. So many tabs open. So many tabs open. Let's see how many tabs I have open. Yeah, yeah, way too many tabs open. Okay, so that's interesting. Okay, take a look at some of that later. Okay, so this nothing. Okay. Um. All right. So I just want to take a little bit of look at this to see what sort of research there might be between epigenetics and genetic variants. So this idea of epigenetics. So most of this idea of genetic inheritance is that you don't inherit the changes to the genome that happen during your parents' life. And that's sort of um, the contemporary understanding of um, of genetic inheritance, or at least the way I understand it. In the past, there was this idea of Lamarckian inheritance, which is the situation where, yes, you can inherit things that happen to the parent's organism's body, genetics code, genetic sequences, that you can actually inherit that. And so for a long time, that was anathema. No one wanted to accept that. No one thought that that was possible. No one thought that that was true. But now epigenetics suggests that maybe it's actually possible um, for for some of these things, right? So and mostly it's done not necessarily through the changes to the genetic code, perhaps, but rather changes to um, what types of genes are turned on and off, in how the quantities of particular proteins that are made, and so forth. So, this is something that's okay. So maybe I was wrong a little bit. It's not about actually changing the genetic variant much or changing the genetic sequence, but changing some of the things with methylation or histones and so forth, right? Um, so so maybe there's not really much of a relationship between genetic variants and epigenetics. But let me just take a look here. Just kind of curious. What is this? 315. Okay. Genomic noise. Oh, that's interesting. I've done a lot of work on noise in the past, so this is kind of fascinating. Okay. Mhm. Interesting. Hmm. Okay. Epigenetics. Hmm. It's only in the title. That's interesting. Kind of strange. Er, Um, but maybe we'll keep that. Genetic variants and epigenetic genes. Genetics and epigenetics. What's this? Hmm. Not much information here, but let's just put this on the research list. Kind of interesting. Let me just look at one more page. This might be a rabbit hole that we don't need to go down. Okay, maybe not. Genetic and environmental epidemic. Okay, let's see about this. This could be potentially interesting. Let me search for something about variants. Right, a large portion of the identified variants are not predicted to result in downstream functional changes at the protein level, excuse me, or are located within introns or intergenic regions. So then, yeah, that's exactly the question. Understanding how these variants may be involved in disease is an important area of active investigation. Okay. Genetic variants can be modified by epigenetic marks. Uh-huh. Right. Interesting. Okay. I think we've gone down this rabbit hole long enough. So let me just make some notes of this just for today. So day 603. That's how I formatted it before. No. Okay. Let's see here. Links. And we can say this. Oops. There we go. Yeah, sometimes Vim on my computer is extremely slow. Extremely slow. I just tried to paste some text in there, a link, and oh man, that's taking its time. I think maybe I need to restart things here. Let me just pause. And it's still not done. Or no, maybe I didn't even have anything. Oops. Oh man. Okay, yeah, I really need to restart some things here. This is just a little unbelievable. This is the problem with trying to run Linux on Windows sometimes. Okay, let me just quit out of all of this. Okay, restart this here. Transino Lab, projects, missions, go. Except for this one, I want to go to stream, text, and open up the ticker, and open up a new tab. Projects, missions, events, lab sessions. Oops. Yeah, no, this is right. This is right. Yeah, this is right. Let me open up stream notes. Great. And while I'm at it, I'll just download these PDFs as well. I can upload them to the GitLab as potentially interesting things. Download this as a PDF as well. I'm going to do all the downloading right now. Okay. And then the last one. Oh no, I guess I had a couple of them. View article, view PDF. Save that. And then we can add this to the list as well. Okay, so maybe this is the best thing to add right now. Let's hope that this is a little bit faster. Not really. Oh, shoot. Oh, so I already have that open there. Let's do this. Maybe I actually just need to reboot my computer. I will do that later during one of the breaks. Okay. And then the last one, which is quite interesting sounding. I'll have to look at that later. Oops. Okay, that's fine. Okay, so we've got these links and everything saved for posterity. All right. Let's see what else we can begin to work on now. Let me also just make note in here of the GitHub that I'm working on, my projects. Let's see here. This is the new GitHub homepage. I haven't been to this for a while. Okay, my repositories. OpenWebRx. All right, let's do this, and let's put this down here as a forked version of OpenWebRx, and let's download this too. Let's make sure I have this set up. Copy that so I can clone it. Let me put it in. Development git clone. Oh, I think that's the old one. So let me get rid of it. Git clone. So this will take a little bit because, yeah, my internet connection here is not the best. Okay, so let's go back to this. So let me open up some of these files here while I'm waiting for that to download. Let's copy that over actually to a day 3. Okay, we're done there. So here you can see, yeah, so this is the OpenWebRx software, which you will maybe play with a little bit. I should actually take a look at that and see how that's doing right now. But first off, I wanted to copy over in TransUnilab projects. So we have missions. Go to the thing here, events, lab sessions, make their day 03, copy day 02, because I'm referring to BCF to day 003. Okay, great. So that's going to be all there right now. We should also put in the texts that I downloaded. So let's make a new directory and say materials, and then I can copy that over. I know that this is so interesting to watch, but this is part of the work. This is definitely part of the work. Sessions, day 3, materials. Great. So that's all there. Cool. Then where was I? I was looking at OpenWebRx. So that's all there. So yeah. So here there's sort of all of the HTML that we can begin to edit and everything. And in terms of how it looks, in terms of how we're going to mess with things and so forth. So let's just check on it. Let's just see how the web interface is looking right now. And yeah, we'll go from there. Let's hope I can get on. All right, I'm able to get on. You know, I am always happy when this happens. I am always so happy when things continue to work like they're supposed to. Okay, let me quickly sort of try and zoom in on this. Come on, zoom in, zoom in. This is some of the tricky bits of working on things remotely on a very bad internet connection. Is yeah, sometimes things take a bit of time to load. Are we going to zoom in? Because I need to zoom in to be able to change this. Okay, there, finally. Maybe? No, it's really not getting it. Shoot. I need to maybe. Oh, no, that's still not doing it. Oh, shoot. It went way over there. Okay, that's interesting. Let's move over there. Kind of curious where my web browser went to on here. Can I open up things? I may need to get someone there to give me some access because I'm certainly having some issues here. It doesn't help that my internet connection speed is so slow here. But I guess while we're looking at this, we could see some very interesting things here, right? This sort of almost spiral that's going down. I mean, that's quite impressive. I've never sort of seen it represented that way. Let me take a screenshot of that. That's cool. That's cool. Um, that's that's just cool looking. I'm sorry. So what that actually is probably um, probably a STANAG or something like that, or some other sort of digital communication mode like that. That that sort of is used oftentimes for different militaries for encrypted communications. So let's write this as spiral. Cool. Okay. Wow. I am just having a terrible time. Let me just try and do this. Come on, zoom out totally. Please just let me zoom out totally. Ah, crap. Okay. I'm gonna have to ask someone there to do this for me because for some reason, just not having any luck with it. Even though I can sort of move this around everywhere. Yeah, okay, this is weird then. So what's going on? Can I close this? I think maybe we need a reboot here. Okay. Well, I'm gonna try something. I'm gonna try and send Control Alt L to try and reboot it. Let's see what happens. Uh-huh. Okay. Oh, I got this. I got the task manager. Oh, good, 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 good. So now I gotta. Oh, there's multiple windows open. That's maybe one of the reasons why. Okay, let's just try and open up a terminal window. And yeah, open up a terminal window, please. I'm asking you, please. Be nice. Oh man. Yeah, see, that's the problem. This is running on a Raspberry Pi 4, and this web interface is a little bit, yeah, resource intensive, and so it's a little bit tricky. And so if you're opening up multiple windows here, then we've got some issue. Let me just quickly message them to say that I need it's rebooted. Let me just do that right now. I'm going to mute my mic while I do this. I'll be right back. Oops. Okay, yeah, something is really up. Let me. Okay, I'm back. So while we're trying to get that taken care of, I'm going to close that out for now. I also wanted to show you. Oh man, now I've lost my mouse. Oh dear. Oh dear. That's not good. That's not good. BNC, why did you make me lose my mouse? Oh man. Okay, I mean, you actually need to reboot because I can't work without seeing my mouse pointer. I just wanted to show you one thing here, which is PSK recorder, which would be showing the different signals that this antenna has been able to receive using this digital mode FT8 over the past 24 hours. And as you can see, quite a lot of them and quite far away, actually, all the way to South America, which is quite incredible. But maybe not so surprising because right now there's this thing called the gray line going on. And, excuse me. And so that's the division between night and day. And radio signals can actually transmit quite effectively and efficiently over the gray line. And so you can actually go all the way to the other side of the planet by by the signals being captured in the gray line. So, actually, I am going to reboot. We're going to come back to this. I want to talk about this a little bit more because this will lead into some of the radio things we're going to do. And then we'll get back to actually working on developing some sort of representation of things. And I'm going to try and figure out how to shut down things um, while I can't see my mouse pointer. So let me, oh man. Okay, I could just see the shadow of it. Let's go over here. Okay, I am going to try and, well, I'm just going to have to log out here, and then I'll be back on the live stream in about 5 to 10 minutes. Okay, all right, I'll be back.
Recording is on.
All right, I'm back. I have a mouse pointer again, which is great. I can actually do my work. Okay, I'm just going to take a quick break, maybe make some more coffee, and then be right back on. Okay, just a moment.
Is on.
Okay, I guess we weren't live streaming. That's okay. So that one is not found in the database. So we'll also add that here. This is CDH8. CDH8. Okay, next. CTNNA2. Let me check my messages here. CTNNA2. 8013. 8013. 6894. 6894. C to a T. C to a T. Overexpression of alpha and catch numbers. Okay. Interesting. And also an allele count of 1. I should perhaps also be saving the links to these. Oh man, so many things to remember when you're when you're not exactly sure what might be important later or not. CTNNA2. PNG, and let me just turn over here. CTNNA2, 1, and then we'll just do that. And that. Okay. Next, I'll have to go back and do those other ones later. DS camel 1. That's interesting. Is this one not the Nomad database? Oh, no, there it is. Just running a bit slow. So we're getting to be about halfway through. 1173 40406 73 C to a G, G to an A. Okay, not exactly the same. So we'll mark that down here. Put that in there. EGF. Oh man, Nomad is being slow, or maybe my computer is being slow. My network is being slow. I don't know what it is, but wow, this is... Come on. Really? Hmm. Unable to get results. Oh my goodness. Do I just really have like super slow internet access right now? Or is Nomad really failing on us? Oh, there we go. So it came up. It's just being a little bit slow today. Maybe it doesn't look like all of my searching, which is really not that much. But no, it's really slow right now. It's really slow right now. Okay. 1108 90223. Nope, not found. So put that down here in the not found category. Okay. EFDH2. Okay. And we're looking for that. Oh man, Nomad, why do this right now to me? You know, I tend to speak to web pages when they're running really, really slow because, I don't know, maybe they'll listen to me. 1573, no, 1575, 1575, 3774. Nope, not found. So I'll also put that down here. Next, Sinpo. Dendritic spines and long-term potentiation. This is something that I used to do some research on. Well, for a summer. I was in a lab working on um, working on they were researching Fragile X syndrome, and yeah, the question of long-term potentiation and dendritic spines and how many dendritic spines were present. So that was a summer that I spent mostly looking at computers, screens, and counting spines, and being rather, yeah, a little bit bored by that. Um, and then also I did some work at the bench. Um, and you know, it was really the summer where I decided that I didn't want to be a molecular biologist anymore, because I originally wanted to be a molecular biologist to sort of discover the gene. I wanted to try and discover the gene that caused my sister's disease called Rett syndrome, which that gene was then discovered like a year later. Um, but yeah, so I was working on at the bench and doing all the things that you normally do in a molecular biology lab, pipetting, um, running electrophoresis gels, examining gels, washing lots of lab equipment, and so on and so forth. And the thing that bugged me was the fact that I was working with this postdoc, and this postdoc, I could see he was basically doing similar work to what I was doing, and basically, yeah, the same kind of work as a postdoc. And I was like, whoa, really? This is what you still have to do as a postdoc? And then I would see the head of the lab basically never spend any time in the lab because she was just having to write grants all the time and sort of manage things. And so those two extremes, I really did not feel good about my future at that point. And so at that point, I decided to try and move away from molecular biology. And so it took me a bit of time, that first year, year and a half of school after that, to figure things out. And then I sort of happened upon this idea of cognitive neuroscience, which at the time felt like a much more reasonable place to be. So I ended up doing my coursework and research in that area and then going into functional neuroimaging and doing work in face and object perception for a long time. And then eventually getting tired of that because I wanted to study things that were not really amenable to be studied within a scientific framework. Um, and so then I moved into design and then arts and then the humanities and new media theory. And now I'm here again looking at molecular biology. So this is actually about 20 years ago that I was doing some of this work. And so, yeah, it's been interesting to come back to this. I mean, I feel good about it. I feel like I needed about 20 years to be able to feel okay about this um, work and not feel a bit traumatized by it. So yeah, I mean, I'm glad. I'm glad to be back doing it, but yeah, took me took me a while. Let's just say that. Okay, digression over. Let's get back to keeping some of this stuff Simpo. Okay, and then let me copy this, and there's an example of one. Again, for some reason, copying and pasting large amounts of, not even large amounts of text, just a URL into Vim in this bash window and Windows subsystem of Linux is, wow, it's slow. It's slow, so slow. Okay, let's move on. TNN, we only have a few left. And Nomad is finally running faster again, so I'm happy about that. Okay, so we have a few things to look at here. 175.054, 558. Okay, that one was not found. What about the other one? 175.04, 6693. Ah, this one is found. Okay, an E to a G, an E to a G. Okay, let's take a look at this. Also an allele count of 1. European non-Finnish male. Okay, maybe I'll go back and also write down something about their sex, because that's also kind of interesting. Because if perhaps these are related to different types of transgender people, then maybe there's something, yeah, wrong also about these hexes. Yeah. 1. Save. Okay. Next. Next. So we have 1, 2, 3, 1, 2, 3, 4, 5, 6. Cool. Oops. No bad. There we go. 4, 1, 1, 0, 7, 4, 0, 5, T to a C, A to a G. So not exactly right. So we'll move that one down here. Okay. And rims 4. Interesting. Long stretches here. Not like that other one. 41107. 41107. Oh, not at all. Okay. 4. Cool. Next is grin 1. Glutamate ionotropic receptor NMDA type subunit 1. Okay, 1400 365 49 C to a T, C to a G. So not exactly the same. So we'll put that down. Again, I'm not exactly sure that this is going to be useful data, but that's the thing with data mining. You sometimes have to just collect a whole bunch of stuff and then decide whether it works or doesn't work and so forth. And so that's what we're going to do right now. Okay, 394 99479. Nope, not found. Map K3, map 4K3. Oops, K3. Yeah, so this is like the this is a kinase. Mitogen activated protein kinase 3. That's interesting. Is that really its name? Yeah, it is. Wow. Okay. Hmm. I mean, that's just kind of interesting. Huh. Huh. You know, the detail that we now know some of this stuff is really quite amazing. Okay, let's finish here. Bulk. Apoptosis regulator. Apoptosis. Cell death. Okay, 242, 50, 1871, not found. So bulk goes down here as well. Bulk. And last one. Da, 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 da. Or is it bulk? It's bulk, not bulk. Oops. Okay. Wow, that's a really long region. 269 50997. Nope, not found. Okay. KCNK3. Yep. So what's interesting is most of these are not found. So they're very specific variants that were specifically found in this study, but then there's a few of them that were, yeah, actually found here. Now, one of the things, I'm just going to go back here and get the links because I think that would be good for us to have in here. 743 83224. Let's just add this here, and then we can do the next one. Now, look, there is probably, let me just search this PPA RGC1B. There's probably a way to do this programmatically, but rather than try and spend all of my time figuring out how to do that programmatically, I decided to do it mostly by hand here, just because figuring out the AKR1C3, figuring out the software libraries for this is I really would need to sit down with someone who could kind of show me how to even begin to search for something like this using the software packages that are available, because there's just like so much possibilities within these packages and libraries, and mostly for things that I don't really want to do in terms of like population studies or things like that. Oh, wait a minute, this one not found? No. 376183661. So CDK2, CDK2, CDK12. Sorry. CDK12. And it's 376. Oops. 376. 18361. 8361. Like, for example, I have no idea where to begin searching for within sort of like a Python library that might interface with Nomad to allow me to basically search like I'm doing right now and find the variant and then programmatically output all this stuff. I just have no idea how to do this. Ideally, that's the way I would be doing it rather than doing this by hand, because it's a lot quicker. But yeah, just absolutely no idea. So what does this mean? It means that the study really identified some rare variants that are only found in a transgender cohort, or at least this is how I would understand things. And up here, I can write, what does this mean? To me, it means that these variants might be found in people who are also not transgender, but it is also possible that these people are transgender, but that information and Okay, looks like it was rebooted. So let me check the BNC and get this thing going again. And I'm just going to mute while I drink some coffee and water. Okay, everyone. It seems to be back, but I'm having problems connecting to the actual um, server. So let me take a look here at what might be going on. Ifconfig, oops, sbin, ifconfig, 10.1.144.873. So you're going to see me try and do this in real time. 8073. Do I not have internet access? No, I do. Okay. Let me. Yeah, there's some weird problem here where I have to actually connect to the port. And then I'm able to get in. Okay, great. So around 20 meters, I'm going to, oh man. Oh, great. I can actually zoom in. Woohoo, this is great. So let me get back here to this so that this looks good for the people in Taiwan. And just to be clear about this, this is not software that I wrote, but it's software that I'm going to be doing some forking of later. And this is these transmissions here are not transmissions that I'm making, but they are transmissions of other people. And so what's interesting here is that you're able to decode these FT8 signals from around the world. So in this screen over here, this sort of zoomed in region, you're seeing these signals. And then the decodings here, which we went over a couple of days ago, maybe the first day, was it? Or maybe it was yesterday. I can't remember. Time is weird for me right now. But what I want to do, because I was getting a little sidetracked with this before, is I want to show you the PSK Reporter website. This is a website used by a lot of amateur radio people to upload the the reception reports that they have of where they're able to hear people in terms of transmissions and so forth. So I was talking before about the gray line, and now that I have my mouse, hopefully you can see this. The gray line here is the division between night and day, and because of the changes in the heating of the atmosphere, that changes the properties of the ionosphere. And shortwave signals rely on the ionosphere to bounce the signals off the ionosphere in order to have some potentially long distance transmissions. So during the gray line, if I'm understanding this correctly, remembering it correctly, the signals can sort of get caught in the gray line and bounce up and down forward all the way around to the other side of the world. So my little antenna in Taiwan was able to hear someone transmitting from Santiago in Chile, which is 18,500 kilometers away from Taiwan. And so it actually probably went the other direction, um, because that's also what can happen with a gray line, is you can sort of go back this way. So 18,795, 19,000? Wow, 19,000. I think that's the record now for me. 19,000 kilometers. So my little itty bitty antenna um, was able to uh, hear someone transmitting all the way from Cordoba in Argentina, 19,000 kilometers away. And this happened yesterday at 10:29 GMT. So this is really impressive. This is really impressive. This is um, more than I could have expected. And so it seems like there's perhaps a good possibility, maybe, that my transmissions later in this week will be able to be heard by my antenna in Taiwan. So um, this is really quite impressive. So what's going on here basically is that web interface that I was showing you, the OpenWebRx, every time it decodes something, let me just pull it up again so you can see maybe a little bit more of what I'm talking about here. Every time it decodes something, um, in this window here with these messages, it uploads that contact to the PSK reporter website. And so that, yeah. And while you may think that that is somewhat obtrusive, that's sort of the whole point of a lot of this amateur radio is to be able to be open about your transmissions. You're not supposed to have anything encrypted on amateur radio frequencies. Um, and there's a lot of um, importance placed upon this idea of um, logging the transmissions that you receive, because there's sort of the sport amateur radio called contesting, which is this attempt to try and sometimes say um, make as many contacts around the world within 24 hours using Morse code, or CW as it's called. Um, there's other things called DXing, which is to try and talk to someone um, in all parts of the world, in every country, let's say, or every county in the US, or so forth. And this sort of leads to these situations where people will then set up amateur radio stations on very remote islands in order to provide that opportunity to other people to mark that one off the map, right? And I don't know. For me, this is somewhat problematic because it often leads to very privileged white people traveling to these very remote places in the world, and um, for the purposes of this very particular hobby, right? And this is not to sort of impugn these people's motives and everything and sort of what they do in those local areas, but it does sometimes seem to me somewhat of a quasi form of colonialism. Uh, and it's not something I'm very keen on. Let's just say that. But there is something kind of interesting here. I'm seeing a fair bit of noise in the signal here. So I'm actually going to try and do something to maybe reduce this a bit. I'm going to keep this open. I'm going to try and do this. I have no idea how well this is going to work in terms of the streaming here. I'm probably like destroying my bit rate, but I am going to change some of the parameters here in terms of the gain. So you'll get to see me change some settings here. So sudo vi config webrx. because what I'm seeing here is that the gain on the software-defined radio might be a little bit too high. And so we may be amplifying more noise than we would like and potentially swamping some signals that we would prefer to be hearing, quote unquote hearing, let's say. Okay, let's go here. And so this is on the 20 meter band. So let me search for 20 meters. And is this the one that I'm doing here? Yeah. So, oh yeah, I need to remind myself here. I need to do one thing. I need to shut down the OpenWebRx service for a bit because I need to get an idea of what are the valid gain values. So when I do this, you'll see the window in the back change because it'll lose connection with the server. So I'm going to say stop, and it loses the connection. Okay, great. So I can do this thing called Rtl Test and get an idea what are the valid gain values. Okay, I'm currently at 38.6. Let's change it down to 33.8 for the gain. Oops, I'm already stopping it. So what was that? That was 33.8. Oops. 33.8. Oops. Oh, right. I already have that open in the other window, the other tab. Okay. Let's change this to 33.8 and system control restart. So that should be restarting. Let me go over here, try rebooting. Hope that this works. Okay, yep, we're connected. Oh, but for some reason, it put it all the way over to there. That is annoying. Okay, okay, this is looking much better. You see how it's not so yellow? There's not so much stuff going on in terms of the, oh man, that's maybe too tightly zoomed in. But let's do that maybe. Let's look at what we get in the window here. Maybe did I turn the gain down too much? Oh, we'll see. Let's keep this going. I'm going to zoom all the way out so those of you in Taiwan are able to sort of see everything that's going on in this band right now. And then I am also going to figure out how to set my home page. So I think I have to do that in options, home. How do I set home page? They don't even use home pages anymore. Firefox set home page. Start up. How to set the home page. Custom URLs. Okay, that's how you do it. So I'm going to go back here. I'm going to copy this URL because this is the URL that I want this to start at. I'm going to copy it. I'm going to close here. I'm going to go over here. Try to at least. And go down to preferences. Not only is this oh, okay. Interesting. Yes, yes, that's exactly what I want to do. Huh, did it do that automatically? That's kind of smart, Firefox, thank you. Okay, so that should now be the home page. Let me test it. Do I want to test it? Yeah, I want to test it. Let's close that down. start up Firefox again. And let's see if this worked. So we at least have the correct URL. Let's see if it actually starts things up properly. Good. It's starting up the decoding. Awesome. Okay. So we are all set. So that every time we start Firefox now, it goes to this homepage. And then we are golden. We are golden. Great. And then we can also change this to be full screen mode. Although that might have been the problem that I had before, but let's see about doing that. Under more. I don't know. Let's see. What does more bring up? Or maybe I can. Oh yeah. This is an interesting thing. Do you see this line that goes across the whole sort of frequency band? These are sometimes tests of particular kinds of um, over the water radar. And when you are listening to shortwave, if you hear sort of this, um, that's what that usually is. There's a whole sort of fascinating thing that we could do about sort of signals identification and exploring the spectrum, which I think we'll, hold off on here because it's going to be kind of hard to do given the challenges oh, there's another one. Given the challenges of working with this remotely using my internet connection at home here, and also just sort of the slowness of the VNC, that would be something to do in an in-person event. So maybe some other day. Maybe some other day. Let me look at mine here and see. Nope, that's not what I want. What is it? Is it? Does this make it full screen? Ah, there we go. That's what makes it full screen. Okay, let me go here. Go back here, and let's see about making this full screen. Okay, cool. We are now full screen. We are now full screen. Great. So this should be looking nice for you all in Taiwan. Okay, let's see. I'm going to close that down. We've done that. We made these edits over here, so you saw a little bit of the editing of that. Close this down. Okay, so now let's come back. As you can see, I sort of go on some tangents here and keep a lot of things in my head, and not so much in my head, but I mean sort of move between lots of different things very rapidly. So we're done with the radio stuff. I think I'm going to take a brief break um, from the stream, just 5-minute break or so, and then I'll be back on for the last hour or so. I may have to leave a little bit early today because I've got some important things that I need to be at 1 p.m. My time, and it's 11 p.m. now. So I am going to put this on pause. I'm going to come back. We're going to spend some time thinking about, again, this encoding thing. This is sort of the key of the artistic concept for this project. That's why it's not easy to come up with. And so we'll maybe do some brainstorming about that, or I will do some brainstorming and stream it to all of you. So let me just pause it for a moment, and I'll be right back. Whoops. All right, I'm back. I'm back? Yes, I'm back. Um, oh man. Oh, I just realized something. I've been trying to record my stuff here, and oh man, I did not add the mic to this. Oh man, okay. Oops. Woo. Okay, hopefully the stream on YouTube is okay. At least the last part of the stream will be fine. Okay, so for the last 40 minutes or so, because I'm going to have to go off stream early today. Yeah, so for the last 40 minutes or so, let's do some brainstorming about, or I'll do some brainstorming and you'll watch me, I guess, about this encoding and what we might be able to do with it. Because this is sort of the key for the project. I've sort of been looking a lot at the technical things recently, but let's sort of think conceptually about this, or I'll think conceptually about this. Just a reminder as well that the body as trans data discussion is today at 3 p.m. Central European time and 10 or 10 p.m. Taiwan time. And actually, I am just going to quickly post about that on Facebook. I'm going to do this with, you're not going to be able to see it because, yeah, I don't necessarily want you to see Facebook. But I will add this lab body as trans data. Huh. Well, this is interesting. As I'm looking at this, trying to find the actual link to the event, there's an article. Let me just actually turn this on here so you can see this. There's a thing here about lab value changes in transgender females. Huh. Oh, this is actually kind of fascinating. I'm gonna keep this. Let me just actually do that. Okay. This is quite sort of fascinating for some other work of mine, but I'm going to save it in here anyway. Value changes in transgender females. Could potentially be interesting for this. Okay, I'm going to go back to this, so you're not seeing my desktop, and then I'm going to try and find what I need to know for hosting about the body as trans data performance today. Did Telegram start? Yes, it did. Okay. Let me see here. Here's the Facebook group. Is this the Facebook group? Yes, this is the Facebook group. Cool. I will post about this. Let me just quickly post about it. I need to, so many things that I need to find here. So many things. Where's the description of this? Oh my goodness. Okay, I'm gonna have to go to my email. And oh man, more emails. Okay, yes, Google. I understand that there is an important policy changes for Google account storage. My goodness, so many emails about that. Okay, public. Please join me and oops, Amard and Cat from Taiwan. Amard from Slovenia for how do we spell this? Body as trans data. Oops. Body as trans data for the body as trans data discussion at 1500. Oops. As part of forking Piragene in lab, kill lab at C lab. 1500 hours CT, 2200 hours CST. And then let me get the thing here, body as trans data. Let's see if this comes up in my search. Oops, let me go back to the, oh man. And I will end up just reading to you the description here so that this is also in posterity. So here is the Facebook event page with Jitsi and YouTube links. The event will be live streamed to YouTube. And here is the description of here's the description. Oh, well, I'll just paste it and then, oops, oh man. Okay, I need to put this in another. I know you're not seeing anything other than my face and some stuff, but that's okay. I just have to do this right now because my email software put really annoying line breaks and things. I guess it cuts things off at 80 characters, or approximately 80 characters. So I have to move all those line breaks. Okay, great. Copy this over here. Copy the event page. Today. All right, so what's this? Okay, so what exactly is this? So this is the description of the body as transgender event. The Exomeofragmissions project perturbs the tension of the body as information. I want to invite you to a discussion with other transgender and non-binary people about the entanglements between biomolecular hormonal jouissance, searches for bioinformatic markers of transgender identity, juridical orderings of trans being, and semiotic performances. How can our visceral trans non-binary experiences inform a more nuanced understanding of identity in process? How can we deeply understand the complicated pleasure of molecular ingestion alongside our semiotic performative capabilities? And in a world that is simultaneously more fluid and more rigid, how can these experiences manifest a trajectory that swerves elsewhere into an in-between space? This discussion will also incorporate some of the developments of the Exomeofragmissions project that is taking place during the Lab Kill Lab work sessions. Okay, so that's the description of what's going on. And I just need to take care of a couple of things here. Let me just quit Telegram. I just need to pause for a moment, and then I'll be right back. Sorry that there's so much pausing today. There's just having my mic down might help. Okay, I'm back. Had to do some arranging for some events this afternoon. No rest for the weary at this time of year. Okay, so let's get back to this, and let's maybe do some thinking about thinking about what we're going to do. Maybe just do some brainstorming in these last few minutes here. In a certain sense, we didn't get a huge amount done today. I don't exactly know where this day went to, this streaming day went to. But that's okay. Sometimes that happens when you're working on these things. So let's just write something here about some brainstorming about encoding possibilities. Okay. So. Just make some PDFs. Okay. So, I would like something that can convey something about the deep time potentials of the genetic code to encode the experiences of gender minorities in the past when they may not have been minorities. So, insofar as the environmental conditions are probably related to changes in the genetic code, or possibly, let's say possibly related to changes in the genetic code, then these changes may thus be expressed in some of these variants. So to explore this scientifically would be in the realm of a kind of molecular anthropology. And so I want to convey the effect of this within the constraints of how these variants are encoded today in order to fork these formats into new forms and to swerve them, let's say, into something else. I just remember that I need to do one other thing here. For later in the day so that this is actually encoded in here. Okay, cool. So let me go back to this. Sorry, I never get tired of seeing it go off to infinity. I know that's kind of cheesy, but I love it. Okay, so the limitations of the VCF format are quite immense. So let's take a look at this again. So the VCF format has these things about file dates. Well, we could maybe play around something with the file dates. So let's play around something with the file date. to explore something about deep time. I know there's a lot of somethings right now, but that's sort of the way this goes. Sources and references. Think about how to modify sources and references. So we really need to think about what kinds of info fields would we like to have that could suggest new types of infos that would perhaps be more suited to the types of studies we would want to see, or the types of data that are more in tune with the directions of this project. Okay, let's see here. Oops, wrong tab. Okay, there's also these filter fields. Oops, we don't need that right now, or maybe we could. Maybe I'll just put these as bullets. The filter could also be important to talk about how certain types of information are filtered out in these studies, and that should be included. What sorts of filters would we need to create so as to filter particular kinds of data in? Oh my gosh, Vim, why are you being so slow? Okay. And then in terms of the actual variance, we can then correlate the new types of fields we create to particular variance, and maybe create new kinds of, oh, I want to put that in a new one. And then more importantly, we can maybe invent new kinds of transgeno variants that would be more in line with our forking and our desires. QO. Then I want to write, do we need to also create something in FASTA or FASTQ format to convey some of this? Perhaps we'll have to think about that a little bit more. And then maybe do we want to create some kind of topology here of variance, like we were finding in some of the videos about variance. So I actually found a quite interesting video last night, late at night YouTube watching. This is my late at night YouTube watching. Genetic variance. Let me see if I can find it quickly. Yeah, so this is the one that I was watching. This one
Hello.
has...
My name is Sarah Stansberry, and I'm an intern at Geisinger. This presentation was developed by Amy Knight-Johnson.
I'm going to put this in the notes as well as a link we were looking at today. Introduction to variance and nomenclature. Q. Come on, Vim, catch up, please. Great. So this particular video had some good tables of these things and examples, and this is something that we could also develop. Create a table and some examples of it, including what's the word I'm looking for? Syntax. Syntactical representations. Great. And then I really want to spend a little bit more time thinking about this kind of interesting question that I have of this data translation. So we have the data translation of genetic code information, genetic variation information, forked genetic variants, the translation of this into radiophonic space. So what does it mean? What does it mean? What are the implications of translating something about the genetic code into a form that then potentially envelops and infuses the radiophonic space of the Earth. And I don't know, this may not be something that's the connection between these two things may not be entirely apparent. To me, there's this fascinating thing about data translation and, you know, what happens when you do these translations. What does it mean to sort of potentially experience this data in this other very different form? I mean, this is one of the things about about representation, which let's not get into a discussion about representation, but really sort of an understanding of or an exploration of this as a form of making ourselves trans. So let's just write this a bit more. So exploration of this translation as a means of also exploring making ourselves trans. And I think I will have to stop for there today. We're gonna have to end about 10 minutes early today. But I think we've got a good plan for what we're going to try and do starting tomorrow. Maybe the last work day where I'll be in front of the computer doing this kind of streaming. And then maybe on Friday, Saturday, and Sunday, we'll be transmitting. I mean, we'll see how things go tomorrow in terms of writing this stuff. So thank you to all of you, any of you, none of you, who were listening today, anyone who is coming through the gallery today to see what was going on. Yeah, you'll see me here again tomorrow from Ljubljana, and maybe it will also be sunny again tomorrow. So I'm off. Remember, if you want to join us, that there's the Body as Translator discussion today at 3 p.m. Central European Time or 10 p.m. Taiwan time, and you can find the links for that. It'll also be on this YouTube channel, and you can find more about this on Facebook. So that's all for now. Ciao, ciao.